Hello,
Just a minute, sir.
it's not an issue. Okay, I'm
Hello.
here.
Yeah. Always, sir. Please continue.
Okay. Sir, teaching experience more than 30 years. Sir, research experience more than 25 years. He's guided 5 PhD students. His work as a referee for doctoral work in botany at Shivaji University, Kolhapur. His in research aspect, he's published more than 30 national and international research papers in reputed journals. He's attended more than 27 conference, workshop, seminar at national and international level. He's delivered more than 16 lecture on scientific temperament and social awareness. Sir organized more than 10 national and international conference at college level. His work as a resource person in national and international conference. He also worked as a member of editorial and advisor board of several scientific journals. Sir, appointment as a member International Curriculum Designing Board for Science Faculty, State Government, Maharashtra. His appointment as chairman on the Maharashtra State Bureau of Textbook Production and Curriculum Research, Balbharti for Science Faculty. This big personality honored with several awards. Best Principal Award by Savitribai Phule University 2017. Best College Award by Savitribai Phule Pune University 2017. Mahatma Jyotirao Phule Award 2019. National Education Excellence Award 2020 for Best Degree College in Maharashtra. International Conference Organizer Award by Academy of Plant Science India is honored as a APSI event scientist and conference organizer award 2012 and 2013. Letter of appreciation by army authorities for collection of higher amount of flag fund. Best teacher award by social welfare section Bank of Maharashtra 2012. Such as Dr. C.V. Murumkar sir means is a multi-dimensional great all-rounder personality. Today, we all have wonderful opportunity to have an interaction with your lecture. Respected sir, we have heartily welcome you in today's lecture series. Thank you.
Good morning.
Good morning, sir.
Good morning. I humbly request others, please mute your audio so that all others can hear properly. Also, I feel that at the end of my session, there will be an interactive timeout where we can have the discussion on the issues if at all you are having any doubts. Before initiating my lecture, let me tell you one thing, that I will be handling over all the notes and this particular presentation to Dr. Kanade sir, and he will be providing you all the details. So the material which is essential for such type of lecture will be made available to you today in the afternoon. And obviously, you can take note of this lecture because this lecture will be again made available to you in the form of a PPT on YouTube by Dr. Kanade sir. Dear fellow colleagues, it's my pleasure to interact with you. I'm very much happy that you have chosen a novel path of research. I do know a few months back, Honorable Prime Minister of India, he made an announcement about the Atmanirbhar Bharat. Obviously, it doesn't mean anything else but we have to show and we have to prove our originality. The days have passed where we were known in the world as a copywriters. The whole world was working some original research work and we were copying it and pasting it in our theses. Now those days have passed and definitely I'm very much happy that eminent and scholar guys like Dr. Kanade Dr. Savan, Dr. Mali, Dr. Telve, and many others, Dr. Shinde, Dr. Jinturkar sir, and many others, they have paved a new path to go for original research. And I'm very much happy that you have chosen very excellent fields in which you are supposed to contribute. As a part of this curriculum, we are supposed to deal with the PhD course program, and I must thank the Research Center, TC College Baramati, for giving me an opportunity to be among you to share some of my views and ideas. Dear fellow, let me tell you one thing, that I am not a fresh season candidature, but I'm a man from old trunk. In other words, you can say I'm the man from old school. Almost what I learned was 40 years back. But the way I learned, I realized that every day new information is pouring in. And obviously this information is nothing but it's a continuation of what we know in a previous way. And so, if at all we are knowing the past, we can understand in a better way the future also and the present also. I'm very much happy that one of my most favorite topics and the issue of interest has been allotted to me to deliver the lecture for. And today, not only I'll be delivering the lecture, but we'll be going ahead with a journey. And the journey is to understand, to know, to realize the process of evolution. Dear friends, I am categorizing my lecture series in 2 lectures. Today, we'll be talking about the general principles of evolution, what we know till today, and tomorrow, as this topic has been specifically included in botany, that also in spermatophyta, but tomorrow's lecture will be specified with the evolution in angiosperms. Obviously, dear friends, if you kindly permit me, we will go ahead with my presentation now. If at all you are having any difficulties to seeing my presentation, do let me know so that I can correct myself. So, to begin with, what is the evolution? It's a very interesting process, and the evolution can be used as a verb where we are thinking and talking about the process, a mechanism. The evolution can be used as a noun where anything which is progressing, any partate or impartate form which is progressing can be called as evolution. Yes, dear friends, even each and every one of you has also evolved, because the environment which was present at your early days, at your childhood, was completely different. And now you have come with this journey of more than 25 years, and today's environment is totally different. You yourself are totally different in and out. Definitely after 25 years, the environment will be again different and you will be evolving again. So evolution is a continuous process. And because of that, it is rather very difficult to trace this path, to trace this mechanism, to understand this mechanism, because it is the mechanism which is indicating not only the journey of an organism, how it is changing without it changing its basic format, but also he is adapting successfully and going ahead. and how at the same time it is coping with the environment. The environment which is present around him, around her, or around every organism. When we are talking about the evolution, from scientific point of view, evolution is nothing but it is a change in the heritable characteristics of the biological populations over successive generations. This sentence looks very simple, but it is most complicated. Because in heritable characteristics, on one side we believe that the organisms which are present, they are present in the populations. And these populations are obviously truly breeding populations. Means the offsprings are very similar to their parents. Their offsprings are very similar to their parents, and it goes on. So when we are talking about the evolution, it is not the process of the progeny, it is a process of genetic diversity tree, not for 1 or 2 or 3 generations, but here we are tracing hundreds of generations. And at the same time, when we talk about this, we have to realize that this continuity of breeding self in sort of an inbreeding of the population, it is somewhere it is broken and new characters are acquired, and even these new characters now again continue on their own for the sexual production or self-reproduction. And because of that, ultimately from a parental form, what we get after successive generations is a totally new genome, new organism, new form, which is a totally many of the times different than their parental forms. When we talk about the evolution, it is not the process which can be counted in years. The years which we know, 1 year or 2 year, 3 year like this. This has to be counted over a process of time. And when we talk about the time, we have to talk about not only hundreds of years, but we have to talk about the thousands of years. We have to talk about the millions of years, and we have to talk about the billions of years. Obviously, there is one more very interesting definition, and that the descent of modifications, evolution is nothing but it is a descent of modifications from the pre-existing species. That is, if the species are present which are truly breeding, then the descent will come, and this descent will be having the accumulated inherited change. So this change will be accumulative. That is, it will be showing not only by the one organism, but that character, the change will be shown by a population, a group. And that's why when a character is shown by a group, it is called a cumulative character, and that is inherited further. And this change is taking place in the population through the time. So obviously there was a parental population, there was an ancestral population, and this ancestral population breeding through in time, they have modified, they have changed, and they have found a new species, and this new species is also breeding through. And this whole process is taking place through time. Obviously, we are leading toward the appearance of a totally new form. And when we talk about this new form, we say that evolution has taken place. Dear friends, let me tell you one thing, that you are the postgraduate students who have completed your post-graduation in flying colors. And so, henceforth, my journey will be not only to teach you, but we will be making a journey with the interaction of what has been inculcated, what has been introduced, what has been added still so far in the process of evolution. And so my lecture will be in the form of an overview. So obviously for the details, you have to rely on the books, you have to go ahead with the notes, you have to go ahead with getting references from the net and so on and so forth. So as per the evolution of the earth is concerned, or the evolution what we know is concerned, dear friends, we are having our own intellect limitation. In the sense that in last 100 years, we were not knowing anything beyond earth. But after 18th century, we started realizing that there is also a world which is beyond earth and what we know as a solar system. Obviously, the solar system is itself present in the Milky Way galaxy. The Milky Way galaxy itself is present in the local cluster, and the local cluster itself is present in a single universe, what we know today. And now the concept has been added in 2003 that there is no any single universe as such, but there are multiple universes. There are many universes and they are existing today. So the journey is from the micro to macro. But we have to restrict ourselves not to the journey of astronomical details, but we have to restrict ourselves what is happening, what happened on the earth. Because of that, as per the evolution is concerned, dear friends, we have to split our lecture into different components. Obviously, the first component is nothing but it's the origin of earth. We must know how this earth originated. Obviously, this was the physical world which was created, because this was the physical world which created an environment. And this environment made the suitability so as to generate the life. So obviously, after the origin of life, origin of earth, we have to go a step ahead, and then we have to trace the origin of life. When we are talking about the origin of life, at that time, we must know what is the definition of life. And then, obviously, once we know what is the definition of life, at that time, we have to initiate with the starting point, starting material, and we have to see how from the starting material, starting point, the today's diverse world has come up. Because we do know that today's diverse world, it contains almost more than 85 million different species. These 85 million different species, they are truly breeding, they are having their own habitat, they are having their own cooperation, and they are having their own competition. What has been rightly said, that every life is giving the food for the other life, and every life is becoming the food of other life. And because of that, when we start with the origin of earth and origin of life, remember, we have to define what is meant by physical world and biological world. In simple words, the physical world is an innate world, it's an inorganic world, where all the components are present in the inorganic form, in the form of their chemical composition. For example, there are oxidates, there are reducing compounds, there are phosphates, sulfates, oxides, and many others, and they are remaining as it is time moral. Means the nitrogen gas which was present almost at the beginning of the earth, even today it is present as it is. The carbon dioxide which was present initially, it is present today also as it is. The silica which was present at the beginning of the earth, today also it is present as it is. And because of the organic world, in other words, it is nothing but the inorganic world. And obviously we have to go ahead one step and we have to see what is meant by, what is the definition of life. And dear friends, life is nothing but, it is the world of carbon. What we are knowing that the life is nothing but, or the biological world is nothing but, it is a life of carbon. What is the difference between the inorganic world and carbon? The difference is that the carbon compound is having the potentiality to rearrange, reshape, remodel, and rejuvenate itself into different forms, taking into consideration all other inorganic material, and at the same time, it is having one more basic important characteristic feature, that it can self-assess and it can self-reproduce. So any carbon which is present, for example, glucose molecule, in the course of a time, the glucose molecule was formed from the carbon dioxide. The carbon dioxide got converted by this carbon cycle into glucose. Glucose was again oxidized to CO2, and again this CO2 was converted back into the glucose. This whole process of modifying, altering, and changing the shapes and giving rise to new assessment, this is called the biological world. And so, if at all we want to define the life is nothing but is a carbon world which is having the capability of replication, reproducing, dividing, and forming the new. And this, how it happened, that particular journey, how it took place, that particular whole process, this is known, this mechanism is known as an organic evolution. When we talk about the origin of life, again we have to think what is meant by earth, how it originated, and what is the origin of life. Now, dear friends, still today we are not knowing the answers for these two questions. First of all, how the earth originated, for this purpose we have to find out the evidences not on our earth, not on our solar system, but we have to find out the evidences far away from us in a different galaxies. We have to find out in the space how the new activities are taking place. Now the celestial clouds are taking place, how the black holes are forming their own shapes, how the red dwarfs are getting formed. That is how the star formation is taking place. And actually the formation of earth is nothing but it was a phylogenetic process in the formation of a star. The earth was not created independently, but the earth was created because of the formation of a Milky Way galaxy. This Milky Way galaxy was formed almost 4.6 billion years back, because it was now called as the Big Bang theory. In this Milky Way galaxy, several particles came out, and these particles, they were having the tremendous energy. This energy obviously got particulated. When it condensed, they formed the stars. And actually one of the middle star, one of the ordinary star in the Milky Way is our sun. So when our sun started cooling down from the gaseous stage, some of its gases, they were left behind because of the gravity, and because of that there was a formation of earth. Dear friends, if you know the solar system, there is very interesting component. What is the component? The planets which are revolving around the sun, they can be categorized into two forms. That is, the inner planets and external planets. So the planets which are present inside the Mars, all these planets, they do have got their own land. So they are solid planets. So Mars is having its own land, the Earth is having its own land, the Venus is having its own land, even the Mercury is also having their own land. On the contrary, the planets which are present beyond the Mars, that is the asteroid belt, all these are gaseous planets. For example, the Jupiter, the Saturn, the Uranus, the Neptune, the Plutos, and their Saturns, all they are made up of gases. They are not having the land at the center. So, in our solar system, there are two kinds of planets are present. And obviously, we are supposed to talk of what happened on the Earth. And because of that, to understand the Earth itself is very important. And once we understand what happens on the Earth, obviously we have to go ahead. Then how come on the Earth life originated? So if we try to trace the earliest records, I told you the Big Bang theory took place somewhere about 4.5 to 4.7 billion years ago. That was the birth date of the Milky Way galaxy. That was the birth date of the local cluster. That was the birth date of the universe. And that was the birth date of the multiple universes. And at the same time, that was the birth date of our solar system. So now, by studying the isotopic forms of strontium, isotopic forms of neonium, and isotopic forms of zirconium, which are spread in the sun, which are present in the other galaxies, which are present in the other star systems, it has been understood that the life of all these isotopic forms is almost the same, and it is 4.7 billion years ago. And that's why studying those, now it has been believed that Earth also was formed at this time. So obviously the Earth was at the beginning, it was a gaseous form. All the components were present in the gas form. and they were too hot. And that's why they started interacting with each other. At the same time, they were revolving around the solar system, and also they were going away from the solar system. But the moment the sun started cooling down, its gravitational force also increased, and all these planets which were present far away, they started coming down, condensing towards the solar system. This whole process of movement of Earth towards the Earth, it took place almost 1.4 billion years. And then what happened? These gaseous forms, they got condensed and they formed the Earth's crust. What we know that today's Earth's surface. So the first earliest known rocks which are present on the Earth in different areas, they are from 3.3 to 3.6 billion years ago. Dear friends, there is one more very interesting issue. That issue is related with the life of or the age of water. Very interestingly, it has been found that the oldest age of the water is recorded, and that is about 2.4 billion years ago. It means after the origin of Earth, what firstly formed was the combination of hydrogen and oxygen in the air, and they formed the water body. And this water, the ancient water, the first water which came down on the Earth, it was created into the form of a sea. And from the sea we have the oceans. And then by reacting with the innate inorganic components in the atmosphere, because of the oxidation and reduction, the first rocks were formed. Remember, the rock is nothing but it is the oxide form, phosphate form, carbonate form of the inorganic components. So when we talk about the rocks, the rock is nothing but these are the chemicals which are condensed together. Obviously, they had the early reducing environment, and this early reducing environment was having the nitrogen, it was having the water vapor, it was having the hydrogen, it was having the helium, it was having the even methane, it was having the ammonia, and it was having some gases like CO2 and even oxygen to some extent. And that's why, as it was very much rich in methane, ammonia, hydrogen, helium, and nitrogen, this was called as an early reducing environment. So this is a geological time scale. Now this is the image you can see at the bottom, 4.6 billion years. The year is called the Precambrian year. It has been found that at 4,000 billion years ago, the oldest rocks which are recorded, even those rocks have shown the presence of early bacteria. Even those rocks have shown the signs of presence of oxidation process. Obviously, this oxidation process must be coming from the photosynthetic bacteria as well as photosynthetic algae. It means at the earliest moment, the plants were having the heterotrophic mechanism. Heterotrophic mechanism means the mechanism where the chemical reactions took place and with which the carbon was recycling ahead. Obviously, after that, the first multi-celled organisms were generated almost 2.5 billion years back. And the firstly, life originated after almost more than 1.5 billion years back. Dear friends, our actual history of organic evolution, it began somewhere about 3.6 billion years when the first ancestral life originated. Then from them, the first 560 million years back, obviously 4.2 billion years back, and there's a formation of obviously land and the early land organisms. And what we know today organisms, what we know as multicellular organisms, this whole journey is of 6,500 billion years ago's process, and we have come today. To make the thing simple, I am having this another chart. So you can see the hidden chart where the work was inorganic. It was followed by the Archean econ, which was the first earliest known ancestors of bacteria, followed by the proterozoic form, where the bacteria and algae they originated, but they were heterotrophic. And from that, obviously, there was a formation of multicellular organisms. So pre-Terozoic era, somewhere about 2.6 billion years ago, is the period where the first life originated in the multicellular form, which were having the true eunucleate structure. Because in archaean structure, we were having the pronucleate structure, but after that we have the eunucleate structure. And then actually the diversification, the biodiversity explosion of what we know as organic form, it took place in known as a Paleozoic period. That also in Cambrian period, almost 500 million years ago, the first land plants, the first land animals were recorded. And then from Ordovician, Silurian, Devonian, Carboniferous, the diversity, the biodiversity of plant and animal life initiated. Obviously from Paleozoic to Mesozoic, from Mesozoic to Cenozoic to present day, we are tracing this geological time scale, that is the age of the different rocks, the impressions which are present in the different rocks, and their own organic and inorganic evolution which has been recorded. Dear friends, let me tell you one thing, that during this whole process of origin of Earth, 13 times on the Earth there was a mass extinction. That is almost 95% of the species, living species, which were present at that time, they became extinct. They were totally removed, they were totally eradicated, and again, every time with the gap of a 20 to 25 million years, the new life originated. So what we know today, what we see today is a completely different scenario than what was present once upon a time in the Paleozoic era, in Mesozoic era, in Cenozoic era. Let me tell you, I will give you two examples. In Paleozoic era, the almost whole land was covered with the very thick forest, and these forests were having the height of almost more than 200 to 500 meters in height. But very thick forest. Unfortunately, there was one asteroid which hit to the earth that time, and the whole land mass was converted. It was inverted, and all these forests, they were buried in the soil. And today, everywhere, including the ocean base, ocean belt, and on the land mass, what we find is the presence of occurrence of the natural gas. We find the presence of coal, and this coal is nothing but it is the decomposed, unoxidized carboniferous material, and that's why we call it as a natural material, petrochemical. So petrochemical material is nothing but it is a hydrochemicals or hydrocarbons, and these hydrocarbons are present in the form of carbon. So the whole forest, they were buried and they got converted into forest. So these luxurious plants, they were completely removed, and what we find in the Mesozoic period were rich in the palm-like plants, in the cycas-like plants, and this is known as a Mesozoic era, which was dominated by the reptiles, which was dominated by the birds. And you will know that the size of the reptiles is almost 100 times more than the reptiles which are present today. And that's why we know the Jurassic Park, we know the Triassic period, we know the Cretaceous period. And then in the Cenozoic period somewhere, we are conquering the world by the most advanced group of mammals, that is mammals. So in Cenozoic period, somewhere in the tertiary period, there was an origin of mammals, and the mammals appeared along with the angiosperms. So the Cenozoic period is the period of, or it is the era of mammals on animal kingdom and angiosperms in the plant kingdom. Remember, even in Cenozoic period, we have seen more than 26 ice ages. 26 times the whole earth was covered with the ice. This ice is appearing on the earth after every 14,000 years. And because of that, if you see the evolution of man, you will find that there are compartments, there are eras, there are components where each component is taking over, changing over after 14,000 years. Today, in Cenozoic period, what we know as a recent period, this recent period is dominated by Homo sapiens, what we know as the wise man. And how it was identified? He was identified by the caveman, which was present 40,000 years back. The caveman, which was present almost 60,000 years back, which was knowing about the fire, which was knowing about using the tools, he suddenly got converted himself into a social animal. And then after 14,000 years, these herd men, they got converted themselves into the farmers. And so after 14,000 years, the basic change took place in the man, and this change was obviously nothing but the formation of a social life, and the hunterer was converted into herderer. They settled down, and the first life of social realm appeared. Obviously, this has been counterparted with the formation of cereals. So the man evolved on one side, and his food also evolved on the other side. And as both are present today, this is the present current world. And with today's lecture in evolution, we are trying to trace what are the components which have taken place and which have played an important role in this whole process of evolution. Now, when we start with, we have to begin about the abiotic synthesis of organic matter. It means it was known earlier that obviously we were not having the carbon dioxide in enough form, because if you consider the organic life which is present today on the earth and total carbon which is present, if at all we release all this carbon in the atmosphere, all the organic carbon which is present in the soil, water, in the atmosphere, the carbon content will be much more larger than the nitrogen content. Today we say that nitrogen is present 78%, but if you consider the all organic life and the carbon which is captured in this organic life, if we release this, once upon a time, this carbon was more in quantity, more in percentage than even nitrogen. And that's why this carbon was present in the form of ammonia, as well as it was present, the carbon was present in the form of methane. Methane and ammonia, these two reducing gases, they came together. The theory has been proposed to give the organic matter. Now, when we are talking about the abiotic synthesis, how this organic matter, how this organic carbon came into existence, actually everyone was telling that this was actually the role of a god. A very interesting experiment was performed by Miller and Urey, and for that purpose, the Miller got the Nobel Prize in 1968. What was the experiment? This is the world famous experiment of Miller. The Miller, he created a reducing environment. That is, he has taken a beaker, and in this beaker, he has taken, you can see at the top, he has taken the methane, he has taken the ammonia, he has taken the hydrogen, he has taken the helium. I'm talking about the top corner of the bulb, and in this bulb, he added these gases. And to these gases, he provided the water vapor from one corner by providing the heat to a beaker, and also he put the two electrodes. Now these two electrodes were charged, and with these electrodes they created the temperature all of a sudden, by the creating electricity of temperature of 30,000 to 40,000 degrees. So in other words, the miller in this bulb, he created a totally ancestral environment, a reducing environment of that time. And obviously you can see what happened was that this water vapor, when it mixed, it got condensed, and in the condenser, the whole water, it came down, and at the bottom, you can see in this beaker, the samples were collected. Very interestingly, it was found that in these samples, they were, when analyzed, it was found that they were containing the sugars, they were containing the amino acids, they were containing the organic acids, and even they were containing the several hydrocarbons. Basically, some of the hydrocarbons, they were of ribulose and ribose sugar precursors. From this experiment, the miller concluded that because of the water vapor and because of this continuous churning of the Earth's ancestral atmosphere, from these inorganic components, the life must have originated. And that's why this was the first experiment which was proved by the miller. Unfortunately, when the many other scientists, they tried to go ahead with this experiment, they could not go beyond certain level of organic compounds. They could not generate the obviously new organic compounds. And because of that, even though the Miller's experiment gave us the idea how the organic materials are generated on the earth, it could not give us the idea how the life originated. And for that purpose, several scientists in 1953, they started about another rule after the discovery of DNA and RNA, and means they realized the complication of the DNA and RNA. The new concept was put forward, and this concept was panspermia. Now this concept was produced by Crick. Obviously, he is the same Crick of Watson and Crick who discovered the DNA model. In 1982, the Crick produced one book, and in the book he said that the life on the earth, it has not originated on the earth, but it has originated somewhere in the space. Particularly at the time, he was pointing towards the satellites which were and asteroids which were supposed to meet us. So these were supposed to meet us, and in English these are called as a comets. So what is said that the comets are rich in carbon, the comets are rich in water, and the comets are also shown the presence of ammonia, amino acids and sugars, and these panspermia, that is these comets, must have brought the life on the earth. And as the earth was having the water, definitely this life then flourished and it spread all over. This particular, his conclusion was based on one finding, and that was the finding which was related with the Halley's comet. It was found that Halley's comet was visiting the earth after every 76 years, and it was noted by studying the history that after every 36 years, there was a large spread of diseases on the earth, the way we are having the today's corona impact. And then he correlated that definitely these comets must be bringing some new bacteria, new viruses, and because of that, they will be spreading the diseases on the earth. And from this, he called this experiment as a panspermia. One step ahead, when this particular Crick's idea was taken up, it was Fried Hoyle, a Swedish astronomer. He started working on this issue, and he tried to find out the evidences for panspermia. His first student was Vikram Singhe from Sri Lanka, and later on in 1982, one student joined him from India, and his name was Jain Narlikar. While he performed one experiment. And what was that experiment? That experiment was repeated at least 4 times. The last experiment was performed in 2012, and the initial experiment was performed in 1992. What was the experiment? The experiment was that they suggested if at all the life has come from the outer atmosphere, outer space, then in the outer space definitely there will be some evidences of bacteria, archibacteria, viruses, or some genetic material. It must be present in that area. So what they have done, that they have said, or they have sent the bubbles. They have not the bubbles, balloons. They have sent the balloons in the atmosphere. The latest balloon has reached in 2012. It has reached almost more than 45 kilometers up in the sky. So it was the area where the satellites are available at the lower belt, because generally the satellites, they move around at about 100 kilometers area from Earth's surface. And they found that some, obviously they collected the earth sample or the air samples from the area, or the samples which were present at the area, and they found that definitely these samples, they contain some of the very interesting fungal spores, bacterial spores, and also some of the viral spores. And the analysis was produced. So this was the experiment which was continued by the Hoyle. And in this way, it was proved, or it is not proved yet, but the whole process of still finding out how the organic life must have originated, it has initiated by the Miller and then followed by Crick and many other workers. So let us go ahead and study the actual theories which have been forwarded in the process of evolution, what we know as organic evolution. So these are the theories of organic evolution. I'm not going to speak much more about this because you are already knowing. Obviously, the theory was proposed by the Lamarck, which is known as the Lamarckism. This is a theory of inheritance of acquired characters. That is, the characters are acquired by one parental form, and the same characters are repeated in their progeny. And in this way, the characters get fixed in that particular organism. So Jean-Baptiste Lamarck, a French biologist, he has postulates that the living organisms and their component parts continuously tend to increase in size. So he said that the evolution is towards one direction, and that is increasing the complexity. The new organs will be produced due to the new need. Need means the environmental changes, alterations, and due to the movement towards the need. So it is the action which is taken by the organism. So he said that 2 roles are there. One role is the environment is changing, and another role is played by the organism to change, to modify, to alter, to suit in this new environment. And because of that, automatically, the new characters are acquired, new forms are acquired, new components are acquired, and these new components which are acquired, then they are settled, they are fixed at the inheritance level, at the genetic level. The development and the degeneration of organisms will be based, obviously, because of that, of the use and disuse. Because if a particular character is acquired because of a certain requirement, if that requirement is not there, obviously the character can be removed, or it will be removed in time. And because of that, his theory was based on the vestigial organs which are present in animals. It was based on the abscission and senescence process mechanism in plants, where he said that these are the unwanted components, and that's why they are thrown out. And because of that, this mechanism itself, that abscission mechanism and senescence mechanism, this has been introduced as a mechanism towards organic evolution. Modifications produced due to above principles are inherited by the offsprings, and such a change will be accumulated in the course of time. So what I said, that in the course of time, these characters will be settled at the genetic level, at the inheritance level, and that's why the new progeny will be continuing with these characters. For this example, he has taken so many characters, and the best example which was studied, discussed, and disputed was of the example of a giraffe. He said that initially the giraffes, they were very similar to the dogs, they were similar to the cats, but when the trees started growing up and when there was a competition, to cope with this competition, the giraffes started elongating their neck. And because of that, today, with the tall trees in Africa, we are finding the giraffes also. And in other parts of the world, where the tall trees are not available, even we cannot find the giraffes also. So this was a theory of Lamarckism. It was having so many questions, so many solutions, so many postulates, so many arguments. But obviously Lamarckism is one of the important components where it was suggested that there is an interaction between the environment and organism. And as a result of these two, the characters are changing. So this is the basis of what the Lamarck has produced. Obviously, we have to go ahead with the Darwin's theory. Darwin was the one which we can say was the most successful biologist, because he was the one, he has given some justifications which were based on the evidences. Obviously, the Darwin's whole process of finding out the truth, finding out the evidences, the geological barriers and the modifications which were found in different birds, plants, animals, all it is already known to us. His historic publication was Origin of Species, in which he claimed that the new species are formed as a result of three basic important principles. He called them as a drives. Because of these three drives, the characters are acquired. Unfortunately, before publishing his data, one scientist from Australia, he sent some of his findings to Darwin. His name was Alfred Wallace. Alfred Wallace, he wrote one article. The article's title was Tendency of the Varieties to Depart Indefinitely from the Original Type. It means he showed and he suggested, because of the environmental force, the parental forms, they modify themselves, and once they modify, these changes they are irreversible. It means the evolution is taking place in only forward direction. Obviously, the moment Darwin came across with these findings and his article, Darwin stopped submitting his submission. He worked together with Wallace. For 3 years they worked together, and then in 1859, Darwin, along with the Wallace, he prepared one paper, and that paper was the theory of natural selection. Now this theory is based on three principles, as I told you, or these are three drives. First, multiplication of the individuals of a species in a geometric proportion. He always said that the species, they always flourish under favorable conditions. That was the meaning of this. And because of this, we are having the largest diversity. So if one species or one formula becomes popular, automatically that particular formula goes on multiplying in a large number, and there is a population explosion. And that's why he told that what we see as a diversity or biodiversity in the world, in flora and fauna, this is simply because of this geometric proportional growth of the populations. Then second is existence of the variations. He said that within the populations also we can find not the homogeneity, म्हणजे एका छापकाने छापल्याप्रमाणे ते तयार होत नाहीत, तर each and every individual of the population itself shows the variations. And these variations are a response of that particular genome with the environment. And because of that, the second character was also equally important, where he was tracing that each and every offspring even though it is truly breeding and it is very similar to the parent, it is not identical to the parent. It is not same to the parent, but this offspring is having certain variations, some changes, some modifications, and these modifications are because of their response to their environment. So the parental environment is different and the offspring environment is different. And most important component was the operation of the natural selection on the existing variability is in order to select the best fitted variation. On the contrary, now the reverse was also true, that all the forms are given a chance to flourish. Unfortunately, some forms they succeed, some variations they succeed, and others do not succeed. And that's why what Darwin has proposed, that it was a chance or it was a principle of random selection. It was a principle of random selection, of a natural selection, which was continuing in this whole process. Obviously, because of that, there was another theory which was put forward, and this theory was called as a mutation theory. The Hugo de Veris, a Dutch botanist, proposed this mutation theory. What he suggested, that in a population, all of a sudden, a new species is generated. And this new species is generated because of certain accidental changes in their genetic material. Or that time he was not knowing about the genetic material. So that's why he used the term heritable change. So what he said, that new species is formed due to sudden heritable change. And this heritable change is permanent. And because of this permanent change, the new species is formed. He found and he studied very extensively the Inothera lamarckiana. And in Inothera lamarckiana, he found that all of a sudden, a new variety was formed. Initially, Inothera was having the pink flowers, and all of a sudden, he found that there was a flower with the blue flowers, and he found that this was a mutation. And then said that this is a mutation theory, and with this mutation theory, new species are generated. A new species reported having the altogether different characters than rest of the population, but they are also breeding true. That is, they are the true inbreeding populations. In the same way, he further also reported the new true extra species of Inothera also. And then from his observations, he put forwarded the mutation theory. So obviously mutation is nothing but it is a sudden change which is reflected at the morphological level. But dear friends, let me tell you one thing. Almost in 90% of the cases, these mutations are vital, and the offsprings which are having them, they do not survive. So we can find the anomalies, but the anomalies, they do not sustain. And they are not part and parcel of the continuous slow successive evolution. However, later it was found that this was the case of a cytological change. That is, it was an issue of inheritance at the genetic level and not because of the point mutation. Because you know well that one gene, what we know as a multiple genes, when the same gene is get to be added, or when the genes are showing the characters like a dominance and recessive characters, they show different morphological traits. So recessive characters show different characters, and dominant ones, they show the another characters. And in this way, the mutation theory may not be possible, may not be successful every time. The mutations, namely chromosomal or gene mutations, they play a vital role for structural changes in the chromosome, that is the ploidy, and also they play a very important role or significant role in the evolution. The incompleteness of the Darwin's natural selection, even though it was used to form a concrete evidence of phylogeny, it was not giving all the rules and regulations. It was not giving the clear picture of evolution. So on one side, we had the Darwin's natural incompleteness, and other side, we had the mutation theory also, which was also having some lacunae. So this led to think that though both are very important in the process of evolution, there are certain other factors as well who are playing a very vital role in the process of evolution. And because of that, several workers, they started thinking in this line. Stebbins, a British taxonomist, actually who was migrated from the Russia, in 1974, he wrote one book, Processes of the Organic Evolution, and he proposed that the following 5 principles are actually involved in organic evolution. So which are these 5 principles? Now, when he was talking about the synthetic theory, no, at that time he was not talking about the principle of organic evolution, but at that time he was talking about the formation of speciation, formation of new species. So because of that, he is concentrating on how the morphological changes are taking place. So what were the 5 principles? Obviously, the first is gene mutation. So the genetic mutations which are taking place at the genetic level, biochemical level, changes in the chromosome structure, that is obviously related with the telomeres, removal of the segments, removal of the sector, removal of arms, and so on and so forth, and the number, ploidy, aneuploidy, diploidy, triploidy, and so on and so forth, genetic recombination and crossing over. Because of that, the new combination of the characters, new combination of traits take place. Obviously, the natural selection, because the progeny which is formed, it has to succeed in the due course of time, and it has to continue, reproduce for its further generations. That is one side. And ultimately, it should have the incompatibility with other species. It is called as a reproductive isolation. That is, it should have its own population, but it should not undergo interbreeding among the other population. That is the reproductive isolation. So the first 3 are responsible for providing the genetic variability. So obviously the basic genetic component segment, and then the remaining 2 are giving the direction for the evolutionary process. Beside these, 2 more are accessory processes. So there are, along with these 5, there are 2 again another processes which are also playing a part, and that is the migration of an individual from one population and hybridization. That is the first component, that is migration of an individual from one population. That is, one form gets changed, altered, modified, and it gets separated out from the population and it develops independently as a species. What has been happened in formation of several new, what we know as a superspecies or varieties or traits. And the second, the interbreeding, backcross and self-cross, which is taking place between the races, between the species, and between the genera, which are providing the additional genetic variability, which is not present in the true reproductive mechanism. So hybridization, obviously, which is taking place between the races, obviously in species and genera, this is called the interbreeding. Now remember, dear friends, such a type of hybridization, such a type of interbreeding took place even in human race. And because of that, because of the hybridization, we accumulated more and more new evolutionary characters, and because of that, we could evolve very fastly. Same has happened even in the family Asteraceae, same has happened even in the family Orchidaceae, and same has happened even in the family Poaceae. And because of that, they are most elastic families of the angiosperms today. Of this, the mutations, genetic recombination, and natural selection are most important process for the formation of speciation. The important contribution in this theory, as I told you, the Crick was the one who wrote and discussed this in detail, but there were other taxonomists also, and they were Dobzhansky, Fisher, Haldren, Wright, Mayer, and of course, a major contribution from the Stebbins, who proposed the museum hypothesis in taxonomy. We'll be studying this issue in detail next time, tomorrow. Now, when we are talking about the formation of species, now, when the theory of evolution has to be proved, now in the modern terms or in the synthetic theory, we have to find out how from the genetic level, from the cytological level, from the morphological level, from the physiological level, from the biochemical level, ultimately a new species is formed. And that's why this is called as a speciation, formation of new species, and the isolation mechanisms. How the species gets isolated from its parental or ancestral population. The differentiation of the populations at the higher levels is taking place at the genera, families, classes, and so on. All available definition of the species stress the importance of the genetic and morphological continuity within the species. It means if we identify a species of a genera, if we identify a genera of a species, they are having characteristic benchmark, what is known as a signature characters. For example, Apocynaceae is having the signature characters, Asteraceae is having the signature characters, the Solanaceae is having the signature characters, Malvaceae is having the signature characters, and all these characters, they are exhibited by their genera and species also. Even though at generic level and species level, they may show several minor modifications, alterations, variations, and that's why they are placed at this hierarchy level of species, genera, family, and classes. So there are several isolation mechanisms. Now these are some of the isolation mechanisms which have been discussed, which are creating the new species, which are creating the new forms. Which are those? The first is a spatial and ecological isolation. I'm not going to discuss this because you are knowing this. This is nothing but the geographical barrier. The mountains, the valleys, the grasslands, even the water body, they are becoming the barriers, and because of that, the one population breeding true, it cannot move from one area to another area, from one place to another place. If at all they want to move, they should have the very effective dispersal mechanism of the pollen grains, fruits, and seeds. And because of that, many of the times, most of the 95% of the species all over the world, in plants and animals, they are always ecologically endemic. They are present in a given ecological niche or area, and that's why they are called spatial or ecological isolation. Obviously, there is second form, that the species are modified as per the environment. There is season, and that's why this is called a seasonal isolation. You do know well that vernalization, phototrophism, all these are playing a very vital role in the flowering recycle of vegetative and reproductive phase. So the long day plants and short day plants, the temperature and the cooling effects, they are responsible for triggering the reproduction in the plants, and because of that the species are different. So you do know well that the plants which are flowering in the rainy season, they are not present in the winter season. The plants which flower in the winter season, they are not present in the summer season, and the plants which are present in the summer season, or the flowers which are present in summer season, they do not flower in other seasons. And because of that, the species, they get varied because of this, what is known as a seasonal incompatibility. Along with that, we have to go ahead, and this is true with even the pollen grains, it is true with the spores, but generally it is true with the animals. And it is called the ethological isolation. And it is called the art of the courtship, or it is competition within the species. What does it mean? You do know well, in birds, there is only one female bird and there are many male birds. You know that in animals, there is only one female and there are many male animals. So in order to attract that female, each and every male, it acquires certain different characters. He may change his voice, it may change his color, it may change its pressure or vigor, and because of that, it tries to copulate with the females. And because of this is called the ethological isolation. So the bird singing, the movement of the leptides, the movement of the mammals, the attraction process, the scents, the pheromones, all these are nothing but these are the ethological isolations. And lastly, the mechanical isolations. Obviously, these are nothing but These are the specific morphological or structural components so as to breed true and so as to not allow other species, other genera, other members to undergo fertilization. So this is what we know as a structural incompatibility. And because of that, the structure of each and every flower is separate. The protandry and protogyne is separate. The size of the style is separate. The length of the pollen tube is separate. The duration of generation of the pollen grain, germination of the pollen grain time is separate, and that is going to create the structural compatibility. And because of all these barriers, the new species, they get isolated from the parental forms and they develop on their own. They develop separate, and that's why accordingly from the parent form, the new species are developed. So this is the allopatric speciation, you can see, which is shown on the left side, and sympatric speciation, which is shown on the other side. So the see at the center what I have written, a single interbreeding population, it gives rise to a geographical isolation between the populations. It leads further to the increased genetic divergence, separation at the genetic level, selection of increased reproductive isolation. That is, within these forms also, the genetic incompatibility, the morphological incompatibility is introduced, and because of that, the totally new species are formed, even though they are present at the same area, same locality, same ecological system, both are breeding independently and both are breeding true. So allopatric, so within the species, and sympatric, the modifications. You can say the new geographical zone is going to create the two species, whereas the new ecological zone is going to create the sympatric speciation. The same is true here. You can see the isolation, which is separated by the barriers, and with this time, the species are formed. So the isolation by the barriers, this barrier may be obviously geographical barrier. Now to undergo changes or variations, the mutations are going to take place. Within the mutations, the next force will be the natural selection. Because of that natural selection, obviously, this mutation, it starts producing true, breeding true, and then it gets separated from the parental form, and we can have the two species separated. Many of the times when these two barriers are, if at all, removed, these two species, they may grow together as a hybrid population, but they do not get converted back to the parental or ancestral population. So it is a one-sided process, it is a forward process, and it is not a backward process. The same thing is shown. So this is related with the gene form at the genetic level, how this is called a cladistics at cytological level. How the A, B, C, D, E, F, G, the living species are present with a diverse genetic combination. But if we study them at the genetic level, at the cytological level, we'll find that they are having the common genes. If we study them, common genes, we will find that there are some ancestral genes, and then genes, they are having a common architecture, common frame. That is, they are having the inducer genes or they are having the suppressor genes, and this particular basic component is the same, and that's why this is called as a central dogma. And this central dogma indicates that they have evolved from a common ancestral species. So with time, how the evolution of speciation takes place at the cytological level is shown. The dark ones are obviously the fossil species, and the figures or the markings or categorization is of living species. Then we come to the last component. I will take just 5 to 10 minutes, and this is related with the evidences, how we should find, how we should gather, how we should compare to understand the organic evolution. So the process of organic evolution can be traced through the evidences gathered through different disciplines. Disciplines, obviously, these are nothing but these are the comparative biological studies at the ecological, that is at the species level or the population level or species level or genetic level, or these may be present even at the larger level, that is the studying the genera or a class or a family and so on and so forth. So obviously the most common which we can study with a microscope is the comparative anatomy. So this has been studied very extensively in animals. So homology and analogy, the relationship between the structures having the similarities due to common ancestry. So obviously, dear friends, we do know that our skeleton is almost similar in the birds, in the reptiles, and in the mammals. Even it is somewhat similar even in the fishes. So this homology, this commonness indicates that we are having a common ancestry. Furthermore, the skeleton pattern of the hand, the skeleton pattern of the wings, or the number of bones which are involved, the number of bones which are present in the fins, they also indicate that obviously they have evolved from a common architectural frame, common structural frame. Then the comparative anatomy also gives the, as I told you, what the De Veris has said, the Lamarck has said about the vestigial organs. So dear friends, we are having several vestigial organs with us. So everybody is having the vestigial organs, that is, they are not in use. Just like the appendix which we have, even the ear growth of the external ears in mammals, like the hind limbs which are present in the snakes, and obviously we are having several characters like the oil glands which are present in the, or talluses which are present in the bark, and the vascular tissues in plants. So these are the vestigial organs. The talluses which are present particularly in the pith, they have been very extensively studied by the FAC, and it has been also included as a vestigial organ. Then serial hemology, that is the serial segment, that is the leaf and floral architecture. For example, you know that right from the pteridophytes to angiosperms, the floral structure or the sporous structure is the same. The leaf structure is the same. It is having the leaf base, the abscission zone, it is having the petiole, it is having the architecture of the veins, and then it is having the tissues of photosynthetic and asymmetry zone. So this is a serial homology which is shown by the plant parts, animal parts, as well as the organisms as such. And then adaptive radiation, that is a modification. Even though the basic structure is the same, we do know that leaf character is the same, but the leaf is having of different forms. The lanceolate leaf is different, the obovate leaf is different, succulent leaf is different, the dry leaf is different, sessile leaf is different, petiole leaf is. So architecture is the same, but the modifications are taking place. That's why, because of they are supposed to sustain in the environment. So these are the adaptive radiations. Then the evidences which are coming from the taxonomy. The full hierarchy of essential taxonomic categories obviously is nothing but it is species, genus, order, family, even the class, division, and kingdom. Means that's showing a common character. For example, we see the autotrophic plant, all these levels, they show the autotrophism. An organism is assigned to each of these categories with the similarities and dissimilarities in increasing and descending order. The degree of close relationship becomes higher as we reach towards the lower scale. Means the variation and diversity is more in the species, but it is always less and less at the upper level. For example, in plant kingdom, we place only the plants which are photosynthetic. But at the species level, we can have a large range of variations. Using such information of a tree diagram can be prepared at different hierarchy levels. Means the tree, variation tree at the species level, variation tree at the generic level, species level, family level is different. And this is giving us the idea about how to trace the path of organic evolution within the taxonomical level. So this is used nowadays to study the phylogenetic relationship of the species, of the genera, and of the family. Obviously, this can be compared together. That's the ploidy number in the genera. That is the cytological studies in the genera. They are going to give us the common characters, because within a genera, we can have the telomeres at a specific level. We can have the aneuploidy at a specific level. And also they show the isoenzyme topology. So many of the times the common enzymes, they are having the isoenzymes. For example, if we study the nitrogenase is having the 13 isoenzymes of its own. But basically these 13 isoenzymes, they have come basically from the two isoenzyme patterns which are very true and which are present in symbiotic previous nitrogen fixing algae. So in nitrogen fixing algae, we are having the true nitrogenases, and then in angiosperms, we are having the 13 different isotopes. So all the characters can be traced together. The evidences, as I told you, in speciation, the evidences can be gathered from the geographical distribution so as to understand the organic evolution which is taking place in the species. Evolution which is taking place in a given ecological area. So obviously this gives a very strong clue in favor of evolution. So the biogeographical realms means zones, they are studied, they have been identified on the basis of similarities and dissimilarities. And because of this, the world, the present world is, we can see the organic world. The present organic world is subdivided into biogeographical realms, like 6 zones have been identified. The Palearctic zone, Palearctic zone, that's the old world. Nearctic world, that is the old zone, that is in North America. Ethiopian zone, which is related with Africa. Oriental zone, which is related with the Asia and obviously south of Himalaya. Neotropical zone, which is present in the South and Central America. And then Australian zone, obviously it is a separate continent. So this can be explained with this diagram. You can see very clearly, we are having the equator, and along the equator on both sides, we are having the center of Capricorn on north side and center of Cape of Cancer towards the side. And you can see the zones. Ethiopian zone is the Africa where the human being originated. The Oriental zone is the one where the angiosperms originated. Palearctic zone is the zone which was the basis or beginning of the pteridophytic zone which was generated. Neotropical zone, which is nothing but it is an extension of Africa, and Nearctic zone is nothing but it is an extension of Neotropical zone and European zone together. So this is a totally new world. In Nearctic zone, we are finding more than 20% of the new angiospermic species are present in this zone, whereas almost more than 38% old families of the angiosperms, they are present in the Oriental zone. Obviously, Australian zone is showing a completely independent parallel evolution. And so the species which are present in Australia, the life forms which are present in Australia, the plants and animal life, there is flora and fauna which is present in Australia, they cannot be compared with these 5 zones because they have evolved independently. And with comparing these zones with other zones, we can show that how with the time, with the given environmental area, the organisms can undergo exhibiting the variations at the genetic level and at the morphological level. The way we are talking, the same is true how they are forming the barriers. So this is the ecological zone. Basically, if we take into consideration the ocean. So you can see at the border level, only up to 200 meters, you can see where the vegetation can sustain. And then we can have the benthic zone, which is where only the aquatic life is present. But this is the zone where the phytoplanktons are present, zooplanktons are present, and that's why they're having their own aquatic different life. So the aquatic marine life, they're having their own way, own method of evolution. And this is supported. Remember, the way the land evolution is supported by the environment and obviously the sunlight, role of sunlight and the components of sunlight, that is duration, intensity, density, and angle, and so on. So in a similar way, in ocean, we are having the volcanic zones. The volcanoes are producing the phosphorus, they are producing the sulfur, they are producing the hydrocarbons, they are producing the methane, they are producing the obviously the ammonia, and because of that, it is having a completely new life. So the life which is present at the base of the ocean, they are following a different line of evolution in aquatic evolution, and that's why it's called the abscissal zone. So we have to study the evolution at two levels: terrestrial evolution and water evolution. So you know the succession, how one species is taken over by the another species, and how this is taking place. This is I'm not going to discuss in details. The evidences from the embryology, the several embryological characters have proved to be taxonomic importance in angiosperms especially, and I'm very much proud and interesting that Maheshwari, Paul M. Maheshwari from Lucknow University, he made a greatest contribution in the studying the comparative embryology in angiosperms. Maheshwari, 1958 to 1963, he identified 12 features of special interest, and that is related with the angiosperm embryology. These are number and agreement of loculi, lacrimal chambers in anther, the structure and thicknings of the endothecium, because this is part and parcel of the microsporangia, and the nature of tapetum, because this is a sporogenous tissue of the male gametophyte. So these three components, they are to be studied independently. They show the variations in angiosperms. The mode of quadripartition of the MMC, that is microspore mother cell. That is when the 4 mother cells are formed, which mother cell is supposed to give rise to the pollen grains? Because out of 4, generally only 1 is functional, and remaining 3, they are degenerating. Now, which is functional? The topmost is functional or the basal is functional? That is also going to give us a very important clue. The development and organization of the pollen grain. So it is the architecture of the exine, architecture of the genitive cell, formation of the pollen grains, obviously their sustenance, time and duration, and the hyaline areas, that is the way areas which are splitting over to give rise to the pollen tubes. Development and structure of the ovule, that is female gametophyte. Form and extent of the nucleus, obviously this is nucellus. No, this is nothing but it is a megasporophyte. Origin and extent of the sporogenous tissue in the ovule, obviously we are studying the megasporophytic tissue and the female gametophytic tissue. The time of wall formation in megasporogenesis, so obviously development, ovule development, arrangement of megaspores, again in this arrangement, which particular megaspore, because they are always monosporic. But only one megaspore is taking part in the formation of an embryo sac, and other 3 are diminishing, degenerating. So which megaspore is taking part? Which is the functional one? The top one, the second top, last one, or the central one? That is also giving us a very comparative account. The development of embryo sac, the way it is taking place, it is 8 nucleate, 8 celled, or 4 celled, 6 celled, and so on and so forth. The formation and organization of embryo sac, the pathway of pollen tube, the interval between the pollination and fertilization, how much time it takes from landing to fertilization. The types of endosperm, obviously you don't know the non-endospermic and endospermic type, that is consumption of the endosperm, totally endosperm by the embryo, or non-consumption, and the presence and absence of endosperm haustoria. So obviously these are nothing but attachments of the haustoria to the female gametophytic structure so as to acquire the food material. Form and organization of mature embryo, and very interestingly, apart from this, remember 40% of the angiosperms, they show abnormalities. They are not breeding true, they are not producing the seeds, but the abnormalities are present at the embryological level, like parthenogenesis, apogamy, and polyembryony. So any cell directly giving rise to the embryo. Apogamy means any part apart from the embryo sac is giving rise to the gamete, and parthenogenesis means without a fertilization, the egg nucleus is dividing into duplicating form so as to give the true seed. Now remember, in embryology in India and abroad, almost more than 100 schools, they are working in these lines of research. And some noteworthy examples of this are Paonia, Trapa, and extensive work has been done to identify the phylogeny in Liliaceae, Lorantheaceae, Cyperaceae, Crassulaceae, and so on and so forth. The evidences from the physiology and biochemistry, morphology is a result of role of biochemical compounds and their mechanisms of synthesis and breakdown therein. So nature of protoplasm, so we are having the protoplast at the beginning, and then we are having the prokaryote and eukaryotic cells, the cell organelles, formation of organelles, their relationship, chromosomes, obviously the enzymes for various actions, the hormones, and ultimately in plants, the secondary metabolites. Now that's why this whole study is called the chemotaxonomy. Now I'm not at all going in details of this, because it is very extensively studied in last almost 60 years. So where we find the differences, amino acids, B chains, phenolics, alkaloids, terpenoids, steroids, they have been extensively studied so as to have the comparative account. Now there is one more component, that is the immune serum produces, that is called serology. It produces the antibodies. Now this is the component which has been used in homeopathy, where the animal organism or animal cells, they react to the chemicals which are coming from the plants, and accordingly they are reacting. So this is because of the formation of antibodies, and as a result of the introduction of the foreign particles of the cells, the way they are responding to this. The proteins carrying the useful taxonomic information, that is actually the three-dimensional structure of the proteins is formation, isoenzymes and other things. They are widely used as antigens. The storage proteins, pollen proteins, are widely studied for serological evidences. For example, these are the examples which are used in homeopathy, like delphinium, hydrostasis, aconitum, bromus, adonis, and you know that almost all the medicinal plants in the world, all the medicinal plants which have been reported in Ayurveda, they are reacting, they are interacting with the humans, they are interacting with the organism body through this interactive relationship. So all this study also gives us the idea about the mechanism of evolution. So see, this is the present proteins they have been identified, the myoglobin relationship, alpha chain, gamma chain, delta chain, and beta chain, and their ancestry has been found, and it has been identified that myoglobins, they are having the most ancestral structure, which was found even in archaebacteria today, that is almost 650 million years ago. Now same has been expressed here. You can see the millions of years, the ancestral gene of the globin, how by various process of duplication, transposition, that change in localization, and more duplications, they have come to the today's form. So separate form of alpha and beta clusters in mammals and birds, linked alpha and beta genes in the xenophus, even the simple and single globin chain in leprosy and hagfish, obviously the primitive fishes, and myoglobin, which is present in mammals. In this way, even the protein structure, in this way, even the photosynthetic enzymes, even in this way, the ribisco enzyme has also been studied and traced following its ontogenic evolution. And lastly, this branch of cytology has made the largest contribution in understanding the speciation and formation of the new species. The chromosome number, their morphology, its behavior, function, and structure at meiosis level is a major cytological data of hereditary material to understand the relationships in population and obviously of the course of evolution. Chromosome number, which is relating to the ploidy form, aneuploidy, that is removing of the number, and polyploidy, duplication. They are also playing an important role in formation of speciation, the chromosome morphology, particularly the aberrations which are taking place, and their behavior at the meiosis also plays an important role. Then duplication, depletion, cutting, breakdown, and all these processes, they are present here. And then the cytological variations at the family level, generic level, species level, or even at the infra-species level, that is varietal level. All these are very extensively studied. आज संपूर्ण जगामध्ये हजारो शास्त्रज्ञ याच दिशेनं काम करत असतात, संशोधन करत असतात, आणि त्यामुळे हे संपूर्ण अल्टिमेटली नवीन स्पीसीज कशी तयार होते, नवीन व्हरायटी कशी तयार करतात, किंवा नवीन व्हरायटी आपण तयार कशी करतो याच्यावरती अवलंबून आहे. बिकॉज वी आर नाउ क्रिएटिंग द न्यू स्पीसीज. अल्टिमेटली देयर इज अ होल वर्ल्ड ऑफ सायकोलॉजी can be divided into these two regimes. One is a DNA regime. So all the obviously new biodiversity which is having the DNA and the ancestral forms which were having the RNA, this can be studied independently. Their mode of function by the formation of enzymes, their proteins which are formed, their correlation and localization and formation of structural proteins, that is channels, all it is give us an idea how the course of evolution might have taken place even at the biochemical level. And when we are studying this hereditary material, we should never forget that this hereditary material has to be studied from the nuclear material, which is the most basic one, but the hereditary material at the alpha helical DNA and 70S RNA from the mitochondria and plastid. They are the first evolved hereditary materials in the ancestrals or prokaryotes somewhere about 2,600 billion years ago. So once we see a single alpha DNA, or if we see any gene or any inducer gene on alpha DNA, that gene has evolved almost 3,000 billion years back from today, and still it is existing. And that's why it is rightly way it has been said that evolution, and to understand the evolution, is not the process to study the past, but it is the process to understand the present. Because the present had stood on this concrete basis of the past, because we have taken all these things together with us. So with this, I finished today's overview of evolution, and I hope that you have understood the way I wanted to present the message. Also, I hope that there was no any ambiguity in understanding, and so I will come back to the main screen. And I will be happy if you make your comments in the chat box. The topic is open for the discussion. If at all you are having any questions, you can have the discussion today, or we can have the discussion even at the end tomorrow, because I'm very sorry that I had to take at least more than 40 minutes extra from your lecture. And if at all there are no any queries, we'll stop for the present. I'll be happy if you give your feedback in the chat box. Thank you.